Hi there, welcome back to my YouTube channel. Today we are going to talk about ribosomes. So let's start our topic with introduction. Ribosomes are small, dense organelles responsible for protein synthesis. They translate genetic information from mRNA, that is also called as messenger RNA, into polypeptide chain, that is also called as protein. Now let's talk about structures and parts of ribosomes. Ribosomes are not membrane-bounded organelles and has two subunits, so it has two parts. The first subunit is large subunit, that is 60S in eukaryotes and 50S in prokaryotes. The other subunit is small subunit, that is 40S in eukaryotes and 30S in prokaryotes. So these are the two subunits, or these are the two parts of ribosome. The one is large subunit and other one is small subunit, as you can see in the diagram. Now let's talk about the ribosome's composition. Each subunit or ribosome is made up of ribosomal RNA, that is also called as rRNA, that provides structural supports and catalytic activity. And the protein, it has two main components. The first one is ribosomal RNA and the other one is protein. Protein assists in maintaining structure and function of ribosomes. Now let's talk about size of ribosomes. Ribosomes' complete sizes are: in eukaryotic ribosome it is 80S, that is made up of 60S larger subunit and 40S smaller subunit. And prokaryotic ribosome, if we talk about, it is 70S in size, that is 50S for large subunit and 30S for small subunit. S refers to sweat per unit, indicating sedimentation rate and not additive. So that's the reason 60 plus 40 is 80 and 50 plus 30 is 70. It's not the bad math actually, it's sweat per unit. And this is how it is calculated. So let's talk about types of ribosome based on their location. There are two types: free ribosome and bounded ribosomes. Free ribosomes float in cytoplasm, synthesize protein used inside the cell. While bounded ribosomes are attached to rough endoplasmic reticulum, they make protein for secretion, membranes, or lysosome. So the two types are: first one is free, that is present in cytoplasm, and bounded is attached to rough endoplasmic reticulum. The second thing is free ribosome make protein for the cell itself, while the bounded ribosomes make protein for secretion, membranes, or lysosomes. Now let's talk about the function of ribosomes. Ribosome has the main important function, that is protein synthesis, and it is by the process of translation. It has three steps. First is initiation, in which ribosome bind to messenger RNA. Then the second step in this is elongation, that is transfer RNA brings amino acids which are linked together in a growing chain. And the third step in protein synthesis by ribosome is termination, that is stopping when a stop codon is reached, releasing the completed protein. And that's how it works. And the second function of ribosome is catalytic role. The large subunit has peptidyl transferase activity that forms peptide bond between amino acids. So it has two main functions. The first one is protein synthesis, and the second one is catalytic role during protein synthesis. Now let's talk about the ribosome's biogenesis. In eukaryote, ribosome components are assembled in nucleolus of nucleus and exported to cytoplasm. In prokaryotes, they are assembled directly in the cytoplasm because they do not have any nucleus or nucleolus. So that's the reason they are directly assembled in cytoplasm in prokaryotic cells. Now let's talk about the medical importance of ribosomes. The first importance is that antibiotics, as many target bacterial ribosomes are used without affecting human ones. Their examples are tetracycline and chlormephinicol. And genetic disorders, mutation in ribosomal protein can cause diseases like Raymond black pain anemia, that is a ribosomopathy. So this was all about ribosome. If you like our video, don't forget to like, share, subscribe, and comment. Thank you for watching.